higher. Oh, yeah. Thanks, Mark. So today it's my really great pleasure to introduce Professor David Baker for this year's Perutz Lecture. So David did his PhD with Randy Sheckman in UC Berkeley, and then he took a BART ride to go to UCSF for a postdoc in David Egert's lab. Now, David Baker is now the director of the Institute of Protein Design at the University of Washington and an HHMI investigator. David is really a pioneer in the field of protein design and protein structure prediction. In particular, he contributed the software Rosetta, which now blossomed into this amazing suite of softwares for protein prediction and protein design. And I'm sure that many of us today have used this software. And I think it's really a tribute to David's work along the years that now he and the field are to the point that they can essentially design proteins that will spontaneously fold in an amazing range of different shapes. And I mean, interacting with David, I have the feeling there is absolutely no limit in what those guys can do. And now they are even to the point that they can make proteins that will use energy to do stuff, meaning they can make molecular machines. They can from scratch design binders for protein of interest, or even design new materials that will interact with biological systems in novel ways. And I think this is really transformative for biology. Definitely it has changed the way I'm doing biology, because now we can use protein design to investigate biological systems in completely orthogonal ways, leading to new insights. Now, I think that David is really a visionary, and for me, he's a perfect example of what thinking big means. And I think, every single project of David is a moonshot, and amazingly, he lands nearly all the time, or probably, as far as I can say, all the time. And I'm sure he will release his lecture today will convince you of this, because his work on the coronavirus system has been absolutely out of this world. But to me, David is not really about scale, it's about elegance. And what I'm always amazed by is that because he's not restricted by what biology has to offer, he can always design the simplest and most elegant structure and most elegant protein to address his problems. And that's really the art of a true protein engineer. And I think this is really remarkable. So David's seminal contribution to the field have been recognized by many prizes. So for instance, in 2012, the Biochemical Society awarded him with a Centennial Prize. And more recently, he got the Breakthrough Prize in Life Science. David is also a member of the National Academy of Science and the American Academy of Art and Science. Sorry. Now, before we start, I just have to say that, as you probably have seen, this lecture and the following Q&A section are being recorded. Talking about the Q&A, if you have a question, you should keep it for the end of the talk. And the easiest is that you just raise your hand, and we will just unmute you so you can ask it to David. If you don't have a microphone, you can also type it in the chat, or sorry, in the Q&A, and I will ask it for you. So in a nutshell, thank you, David, so much for accepting our invitation today. We really look forward to it. And without further ado, the stage is yours.
Oops.
Does it work?
Sorry, we tested it just a moment ago, but let's see. I just have to find the right window. Shoot, it will work. Sorry about that. Where did it go? Here it is. Well, thanks. It's wonderful to be here. I wish I was visiting you in person, and I just have to sort of turn around. So that was a very nice introduction from Manu, but we have the feeling that, or we actually have found from experience that Manu can solve any problem in cell biology. So I think we have this wonderful collaboration, and he and his student Joe Watson have just performed miracles with our designed proteins, and it's been absolutely amazing to work with Manu and Joe. And I tried to put in my talk the examples of things that we're doing together, but I realized there were so many that I couldn't possibly fit them in. So So that's been one of the really fun things for me in science the last year or two. Okay, so today I'm going to tell you about what we can now do with de novo protein design. And I'm going to sort of start by framing it in the context of the coronavirus problem. So the basic principle underlying protein design, well, up until very recently, is that proteins fold to their lowest free energy states. Now, this isn't always true, but it's a good, it's a good first principle for organizing thinking. So if you want to predict the structure of a protein from sequence, you search through the different possible states of the protein and evaluate their energies. And the native structure will be the lowest energy structure. If you want to design a protein, you make up a brand new structure, and then you need to find a sequence whose lowest energy structure is that state. And we've developed a computer program, we and our colleagues, called Rosetta over the years, that does the sampling in sequence space and in structure space and evaluates energies. And we've been sort of progressively increasing the Rosetta energy function, trying to describe van der Waals interactions, hydrogen bonding interactions, solvation interactions as accurately as possible. Now, very recently, this whole picture has gotten a bit upended, and we're using deep learning networks to hallucinate new proteins. And if there's time at the end, I'll say a few words about that. So what I'm going to focus on today throughout my talk is the problem of de novo protein design. And this is a very interesting problem because the number of possible sequences, even for 100 residue protein, is astronomical. And nature and the evolutionary process have only sampled a much, much smaller fraction, a very, very tiny fraction of this. So if we think about all of sequence space being represented by this gray bar, then naturally occurring proteins, fall into these clusters, protein families. And what I'm going to be telling you about today is designing new proteins from scratch, from first principles, that are completely unrelated to any naturally occurring protein. So I'm going to tell you about a lot of proteins today. None of them have any sequence relationship to any protein of known structure. And so in all of the everything I'm going to tell you about today, after we do the design calculations, we obtain synthetic genes that encode these new amino acid sequences. We put the synthetic genes into bacteria or yeast. They produce the proteins, and we purify them out and then characterize them. We're also doing a lot of work now going beyond the 20 naturally occurring proteins, 20 naturally occurring amino acids, and making synthetic molecules by chemical synthesis, but I won't be talking about that today. So I'm going to organize this around COVID. So when the pandemic broke out, as you know, the sequence was published more than in January of a year ago. And when from the sequence of the spike protein, we were able to build actually quite a reasonable model of the spike glycoprotein. And then as the cryo-EM structure and crystal structures were determined, we switched over to using that. I'm going to tell you about what we have been doing as far as antivirals, diagnostics, and vaccines. So for antivirals, we have been focusing on blocking the interaction between the spike protein and the ACE2 human host cell receptor. So we've been, it was kind of, the timing was interesting because we've been making a lot of progress recently in designing proteins to bind to protein structures of interest. So basically, without using any information other than the target structure, we can now design binding proteins. And so that's sort of depicted on the right here. So we make up very large libraries of hundreds of thousands of different protein backbones. And then we dock those against the target. In this case, this is the receptor binding domain of the virus. And we identify those docks which are shape complementary and from which we can make chemically complementary interactions with the virus. So we first dock the scaffolds and we design the interface. And we pick out those where the interactions really look optimal. So the other approach we used here was to take a piece of the ACE2 helix. So this is using kind of privileged information because we have the structure of the complex between RBD and ACE2, and then we built small proteins around it. So this one's an ACE2 mimic. These designs sort of cover different binding sites on the surface of the virus. And here are examples of proteins produced designed using these two different methods. So this is one that was designed completely from scratch, and it's 55 amino acids, hyperstable, and it binds with about 100 picomolar affinity. Now, interestingly, the one where we use the ACE2 helix is, first of all, it's a bigger protein because we need to fit the ACE2 helix in, and it's considerably lower affinity because it's based on ACE2. It's closer to 10 nanomolar. So we can actually do better in terms of binding an arbitrary new target by building from scratch. Okay, so this is now the cryo-EM structure of this 55 residue de novo design protein bound to the spike trimer. This is from David Biesler's lab, and you can see one of the RBDs is in the downstate, two are in the upstate, and you can see the protein in ribbon here. Now, what's really exciting from the computational design point of view is if we compare the structure, the experimental structure shown here, with the computational model, which is what we, I described this docking and design process, you can see that the design protein is exactly where it was supposed to be. The backbone is in exactly the right place, not only sort of internally in the fold, but also placed relative to the RBD. So the computational model and the design are superimposed on the RBD, and so you can see the design, the design protein is in exactly the right place. If we zoom in and look at the side chain interactions, we see that those are nearly identical to the design model as well. So this was really our first indication that we could design, well, one of our first indications that we could really pick an arbitrary site on an arbitrary protein scaffold and design very high affinity binders to bind there. Okay, so these proteins are very potent neutralizers of the coronavirus. So this is an experiment where live virus is being added to cells, and then different designs are being titrated in. And the one that I showed you is this magenta line here at about 15 picomolar, which is 0.15 nanogram per mil. This protein gives is blocking entry. That's the IC50. And this is it's a small protein, so this corresponds to just 0.15 nanogram per mil. It's perhaps the most potent compound known to date to block entry, block the ACE2 coronavirus interaction. Now, there's been a lot of, with collaborators, we've been doing a lot of animal experiments. This is with Syrian hamsters, which are one of the classic models for coronavirus infection. So hamsters that get the virus get very sick. It's a very acute infection. And but if this protein is administered intranasally from 48 hours before to 24 hours after, there's almost no effect. You can see that we're getting nearly complete recovery by 7 or 8 days here. So far I've described, I showed you that the small protein can bind to 3 different RBDs on the coronavirus. And so we've obviously been very concerned about escape in the last 6 months. And so we focused on, even though we're at 15 picomolar binding of the individual domain, we thought we could obtain even higher avidity with higher affinity by making constructs which engage multiple RBDs simultaneously. So here what we've done is to design a trimeric version that holds 3 of these binding domains in exactly the right orientation needed to bind to engage 3 of the RBDs. And here's some negative stain EM data showing that the trimer, this design trimer, in fact, sits on top of the spike. We actually have a pretty nice cryo-EM structure of this now. So we can take binding domains and then position them quite precisely. And I'll be alluding to that in a moment about in the context of designing biased and superagonists for cellular receptors. So with these multivalent versions, both trimerized versions like the one that I just showed you, and versions where we take 3 different domains and connect them by linkers, we get very potent protection against all the escape versions of the virus that we've seen. So this is the South Africa and the UK versions, for example. And these are these different multivalent versions. We're again getting IC50s, which are better than about 100 picomolar. we're seeing similar protections in animals as well. Another nice thing about these small proteins is they're not like antibodies. The problem with antibodies, particularly for something like the coronavirus, is you just can't, there's not enough manufacturing capacity to produce them for everybody who would need them. But these small proteins express very well in E. coli. So this is just a whole cell lysate expressing that de novo designed protein. It's down at the bottom, 55 amino acids here. You just heat the cells. The protein, most of the E. coli protein denatures. This is at heating at about 80 degrees. And the only thing that's left in that's soluble after spinning that heated lysate is the designed protein. There's some small levels of contaminants which can be cleared up quickly. So we think this is a way to really, really drop the cost of drugs and coronavirus antivirals in particular. So we are we're sort of going through the steps to get these to clinical trials, and that's been a very interesting. There aren't one of the tricky things has been that there isn't a lot of precedent for the idea is a nasal spray, so you could just have it. You don't have to refrigerate this stuff, so you could just have it in your go to the drugstore, get it sprayed in your nose if you're going to get on a plane, or if you're in India and just in a dangerous place, you could take it when you needed it. And again, it's super stable, so you don't have to keep it refrigerated, and distributing it should be much more easier. And then the other thing that we're working about on this context is it did take us a while, and there was a fair amount of experimental screening that we had to do to find these really, really good designs. So we're trying to now improve this whole computational pipeline so we can go from sort of identification of new pandemic threat to very high affinity binding proteins in just a few weeks, which we think is possible. Now, we've taken the same methodology, or actually in parallel, Longjin Cao and Brian Coventry really made this enormous breakthrough in the last several years, being able to sort of, like I said, take a protein of known structure, pick a region, and then design a very small protein, less than 60 amino acid, that binds to it. And so they've been making binders to the major cell surface receptors like FGF receptor, PDGF receptor, EGF receptor, and then many cytokine receptors. And I'm just showing you a few of them here. And we have crystal structures now, many of them, and again, they're binding very much like they were supposed to. And these are all, it's kind of interesting, they're all very small proteins. This just shows surface views. And they have their shapes and chemical properties, though, are different enough to make them very specific. So this is the different design binders, looking at binding TED experiments against the different targets, and you can see we're getting really good specificity here. In the case of the EGF receptor, we made binders to different receptor subunits. So now what we're very excited about doing is trying to manipulate cell fate by making super agonists and different types of agonists which hold different receptor subunits together in different orientations to get new signaling outputs. And we have there's a first example of this. Where this is from a year or two ago with Chris Garcia, where basically we made scaffolds which held the poietin receptor at different angles and distances from each other. And what we found is we got different signaling outputs. So now with these small binding domains, we think we can really generalize this to signaling. There's all sorts of different exciting applications like transdifferentiating cells in the body for regenerative medicine and other applications. So very excited about that area. Okay, the second area, in response to COVID was diagnostics, developing sensors. So far I've talked about designing these really rigid sort of rock-like binding modules. But for this, we wanted to see if we could build molecular devices. So here we have a molecular device which has two states, has a closed state, which is dark, and an open state, which emits light. And these this opening closing transition is coupled to the free energy of binding a target. So we've got a binding domain that's closed here, and when the target's present, the binding energy drives opening. And so what we've done here is simply take that small coronavirus binder that I described, embed it in the closed state of the system, so that then when the coronavirus is there, it opens up and light is reconstituted, light is emitted. Basically, luciferase gets reconstituted. And this works really well. So the sensor is dark, and then when the spike or the RBD is added, we get this very rapid and large increase in luminescence. More recently, we've been exploring this for monitoring responses to vaccination. So since this is in thermodynamic equilibrium, we can have the sensor, we add the RBD and it opens. Now, if we add serum samples that contain antibodies against the RBD, they compete with the sensor and we get a loss in signal. We actually have nicer data than this now, but so we've been looking at samples from patients who have been vaccinated who have different levels of response to the virus, and we can assess their antibody response by looking at the neutralizing antibody titer, and we can also look at the change in signal. You can see they're quite well correlated, and so this is a very quick test you could run at home, read on a cell phone, and so we're hoping this will be useful. We can take the same concept, and I showed you we can design binders to many other types of proteins. And so we can convert these into sensors in the same way by caging them in the off state of the device, so that the protein device, so that when the target is present, the thermodynamics drive opening. And so, for example, this is a troponin sensor. In these experiments, we've got many different sensors, and then we're adding to each one the target. And this is similar to the specificity plot I showed you before. Only when we add a troponin to the troponin sensor do we get the light emitted. And here's, for example, one against the botulinum toxin, where only the botulinum toxin sensor is responding. So we think this is sort of a very general way of making molecular sensors. And again, I wanted to give you an example of how we can use this sort of molecular device concept for problems sort of beyond coronavirus, and in this case, beyond sensing. And here, this is a rather different problem, at least on the face of it, the problem of doing calculations in the body, because antibodies just work by going after specific targets. But sometimes you want to do more subtle things, like you want to recognize cells that have two markers, but not just one alone. So you may have a tumor that doesn't differ from healthy cells in any single marker, but differs in the combination. So we can take the same type of switchable system and direct the different components to different cell surface proteins. And so only when we reconstitute the full system do we get switching. And in this case, rather than emitting light, what we're doing is we're exposing a motif that recruits an effector, for example, a CAR T cell. And that example is shown here. So here, this is a little bit more complicated. So imagine a tumor cell, which has two markers which only occur together on the surface of the tumor, and then also has a healthy cell marker. And we want to make sure that we don't activate CAR T cell signaling of any cells that have that healthy cell marker. So then what we do is we take the two components of the switchable system that I mentioned, that I described earlier, and direct it to the two components which occur on the tumor cell. And then we take a third design, which actually sequesters one of those two switching components required for switching and direct it at the healthy cell marker. And if we do that, then we only get CAR T cell recruitment and activation when in the presence of cells that have the two tumor cell markers, but not the healthy cell marker. So that's here. And the third area that I wanted to tell you about in context of COVID is vaccines. So for a number of years now, we've been developing methods for designing self-assembling proteins, and much of the rest of my talk is going to be about self-assembly. And in particular, we have been developing a wide variety of polyhedral nanoparticles. And so the basic principle is to match the symmetry axes of a polyhedron. For example, in this case, an icosahedron, which has 5-fold and 3-fold symmetry axes, we place protein homoligomers with 5- and 3-fold symmetry on those symmetry axes, and we can move them in and out and rotate them, and then we design interfaces between them. And using this procedure, we've been able to design a wide variety of very homogeneous protein nanoparticles. You just express the two components in E. coli, they self-assemble, and these are just EM fields of what comes out. And so they're very homogeneous and robust. So my colleague Neil King at the Institute for Protein Design has taken the receptor binding domain of the spike and then displayed it on the surface of these one of these nanoparticle systems. And again, he gets very homogeneous nanoparticles. You can see the little RBDs, relatively little on the surface here. Now, what's very exciting is that these induce very, very strong responses. They're much stronger than to the trimer alone. And this is because avidity. We think it's because avidity is really stimulating B cells that have the corresponding B cell receptors. So the current mRNA and other subunit vaccines basically use the trimer, and now with these nanoparticle vaccines, there's considerably more neutralizing antibody elicited and better protection. Oops. So these actually have been in clinical, Neil's team has these in clinical trials for the last 6 weeks, and we're about to get feedback on them. These are among the most promising of what are known as the second generation coronavirus vaccines. So now we've, again, going beyond COVID, we've been sort of extending this concept of building nanoparticles from proteins with symmetry. And what we realized is that we could actually combine form and function by embedding symmetric proteins in these nanoparticles that actually have functions in addition to symmetry. And here we're backsliding a little bit because one of the proteins we're using is actually a naturally occurring protein. I think that's the first time in my talk where that's happened. And so these yellow things are antibodies. And so what we've done is to design, in this case, a pentameric protein which binds the Fc region of an antibody such that the 5-fold axis of the pentamer and the 2-fold axis of the antibody lie on the 5-fold and 2-fold symmetry axes of an icosahedron. So what that means is if you take this designed pentamer and add it to any antibody, it will assemble it into a perfect icosahedral nanoparticle. We've also made C4 proteins tetrameric proteins that hold antibodies so that they're placed on the 2-fold axes of icosahedra. And here we have a trimeric one that holds antibodies so they're on the 2-fold axes of tetrahedra. And so you can take any antibody and rapidly format it in these different oligomeric states. And then you can look at the effects of the antibody, for example, on signaling. And this is an example with an anti-death receptor antibody where we can basically, this antibody alone doesn't do anything to tumor cells, but by changing the presentation of the antibody changes the engagement of the death receptor, and we get killing of the cells. So if you have any antibodies you're interested in and you'd like to potentiate or explore how clustering of their receptors alters what the effect of that is on signaling, now you can do it very easily. There's no covalent modification or anything else required here. It's just non-covalent interaction. So beyond biology, I just wanted to show you sort of kinds of things that we're thinking about. So, and this is happening a lot in the group now. Nate Ennis designed a C2 protein that binds 2 chlorophyll positions, as in the special pair in photosynthesis. And then those have very interesting optical properties on their own. And then Shunji Wang built these C2 structures into octahedra. And so now we have octahedra with a C2 chlorophyll pair on each axis. And this is actually just showing you, again, this is a case where the design is very close to the computational model. So we're now thinking about putting electron acceptors in the middle, like nanoparticles, to try and start creating artificial light harvesting complexes. Okay, so now this is where I start talking about all the fantastic things we've been doing with Manu and his student Joe. So this is work by Ariel Ben-Sasson, and so Ariel took a D2 protein and a D3 protein and arranged them so that they would come together to form a hexagonal lattice. And this is what happens when you produce the proteins in E. coli. You get this very nice lattice, and he put a GFP in at the trimer vertex, and you can see the GFPs there. And so then Ariel made the proteins separately, and when he mixed them together, they spontaneously form this really beautiful and regular hexagonal lattice. And this is a superposition with Ariel's design model. There's the D3 and the D2 components. They're exactly where they're supposed to be. So Ariel, in a great good fortune for my group, met Manu at a meeting a few years ago. And we've had this really exciting collaboration where Joe and Manu have been functionalizing these components and getting really spectacular results, which I'm hoping you've all heard about, so I'm not going to go over them again, in being able to demonstrate assembly on cells and very interestingly, these arrays shut down endocytosis, and they have really profound effects on cell polarity, which if you haven't heard Manu speak about, you definitely should. It's absolutely amazing. So we've also been thinking about in nature we have proteins that interact with inorganic minerals and cause mineralization, as in tooth and bone and seashells. What if we could design proteins that could template inorganic material mineralization? But what if we could do it with semiconductors and other materials that are of interest to modern humans? It would be really cool. So we've been starting to look at this. So this is work from Harley Piles. He designed proteins which are perfectly epitaxially matched to an inorganic surface. So they're repeating proteins that have a repeat spacing that matches, in this case, the mica lattice. And he made these proteins long enough so they were visible by atomic force microscopy. You can see them binding here. And then what he did was design interfaces between these to direct assembly of different types of assemblies. And this is now a hexagonal lattice. Again, it's different from one that Ariel designed because this is only forming on mica. So these individual elements, proteins, are epitaxially matched, and then they have interfaces which direct formation of this basically trimer on the mica. But since these are perfectly straight proteins, Harley could make a series of variants just by making them shorter and shorter and shorter. This doesn't really work with naturally occurring proteins, but you can see the experimental results down here. The hexagonal lattice gets smaller, finer and finer, and these are averages down here. So what we're now trying to do is to go flip this around and use the designed proteins to actually template inorganic mineral growth. Okay, so everything I've told you about so far is soluble proteins, but we can take the same principles and apply them to membrane proteins. And this is work of Chunfu Zhu. He designed these alpha helical channel-like proteins, which have 2 rings of alpha helices surrounding a central pore. These are soluble protein designs, and these are comparing the design models to the crystal structures. So he and Peilong Liu then took these and they resurfaced the outside to make them membrane compatible and expressed them in mammalian cells. And what was very exciting, this in collaboration with Bill Catterall, is that these proteins turned out to be very selective potassium channels. So this is looking at conductance. This is now a patch clamp on a mammalian cell expressing this protein. This is looking at conductance for potassium. You can see it's much more selective for potassium than sodium. And this is kind of interesting. There's been so much written about what makes potassium channels selective for potassium. And here we could just design a protein with a pore and get potassium selectivity pretty much right off the bat. Now, what we can do now is, and we're doing is we can start exploring the effect of the exact composition of the lining on the ion conductance. In this experiment, Chunfu took a glutamate that was near the entrance of the channel and turned it to a cysteine. And what he found then is that, it's easiest saying here, is that this channel still conducted ions just fine, but if he chemically modified that cysteine, the conductance went away, sort of demonstrating that the ions are actually flowing through this central channel. Now, Peilong and Chunfu also, I told you there were those larger channels, the ones with the 16 helix, and those, this is a cryo-EM structure Peilong solved, and there's a really big channel here, which actually allows small organic dyes to go through. So those were alpha helical channels, and Anastasia Vorbieva took on the challenge of designing all transmembrane beta sheet proteins. And these have some very interesting properties. This was a very challenging design problem because it turned out the key that was very unintuitive at the beginning was to make the beta strands extremely low beta sheet propensity, because the design, since they're very hydrophobic, would tend to form amyloid, and she had to really increase the cooperativity by reducing the strength of the interactions. So this shows a crystal structure comparing her design to her design model and a crystal structure, and they're very, very close. Now, what's exciting about this is, as many of you know, these transmembrane beta barrels, the naturally occurring ones, have been used for things like single molecule DNA sequencing and other approaches. Now we can pretty much design them to order. And so here are some of the ones we're currently designing with much larger pores, for example. And we're very excited about using these for, as filters, maybe for various types of sequencing and so forth. Okay, so then the very last part of my talk, I just want to talk about designing, coming back to this idea of responsive designs that aren't just rocks, that actually sense the environment. So I'm going to focus on pH sensitivity for the next couple minutes. So this is a design of Scott Boykin's. This is a C3 trimer where it has these different types of layers. The red layers look like this. They have buried networks. You're looking down from the top, that involve 3 buried histine residues that are fully making hydrogen bonds. These black layers are hydrophobic layers. Now what happens is when the pH drops, this thing is a very stable protein at pH 7. When the pH drops, these histines become protonated and the whole thing blows apart. And Scott could control the pH at which this happens and the steepness of the transition by controlling the number, basically changing the number of these histine layers, and by changing the overall stability of the protein. So the number of histine layers determines the cooperativity, and the overall stability, which can be tuned, basically determines the set point. And this is a crystal structure showing what the hydrophobic layers look like and the these hydrogen bonding layers look like. So again, we can design these very accurately. So this is, again, a repeated theme now in my group. We have these really cool building blocks, this incredibly pH responsive protein, and how Shen took this protein and used it and took it and built it into one-dimensional fibers, basically by docking it against itself and then designing the interface to promote dry assembly into fibers. And these are just cryo-EM pictures of the different types of fibers he was able to make. I won't belabor this, but this it's with Justin Coleman and Eric Lynch, solved the structure of this fiber. And like many of the fibers that Howe has designed, it's nearly identical to the design model. So here we've designed a monomer to be pH sensitive, and then Howe has very precisely arrayed it into a fiber. So this is another example of really cool collaboration with Manu and Joe. So first of all, we had done, what Howe did was just look at different pHs. You have the fiber at a pH 8, you drop to pH 3, and then it when you raise the pH again, it goes back. But we had no idea how fast this happened. This is a really cool experiment of Joe's. This is with fluorescently labeled fiber, and so you can change the pH and follow what happens after time. And you can see that in a very small amount of time, the fiber basically explodes. And so we can build in, it's almost like a 1D phase transition, because this thing is so cooperative. Each unit has 9 histines in it, and then the whole thing is made out of these units. So we're very interested in sort of figuring it, sort of zooming in to see what's happening here. And it's very, very sensitive. So there's no effect, in this case, being at pH 3.4 has no effect on the fiber, but at 3.1 it comes apart. And Howe can change the set point by changing the number of histines inside the monomer. And then the final area is, I think some of you heard this from Alexis Corbet, is now we're starting to think about designing rotary machines. And so what Alexis has done is design proteins which are axles, and then other proteins which are rings, and he's figured out how to get the rings to assemble on the axles. And these are examples. Here's an axle and a ring, and then he's got the ring assembled on the axle. And so he's done this with both in cases where the symmetry is matched. So he has a C3 axle and a C3 rotor, as well as other cases where there's no match at all in the symmetry. So for example, a D3 axle and a C5 rotor. And when you do this, you get very different types of rotational energy landscapes. When you have a match in the symmetry of the axle and the rotor, you get sort of a simple threefold symmetric rotational landscape as evaluated by Rosetta. But when there's a symmetry and a mismatch, it's a much more rugged landscape. And so then what Alexei has been doing is using cryo-EM to map the rotational landscape of these. And I'll focus in on this system. So here's Alexei's computed landscape. And here are different minima, that rotational minima that he observes experimentally. So you can see that in this case, this is a case where there are two rings on the rotor. You can see here they are staggered relative to each other, and here they're eclipsed. So these are, and so we have speculations about what the differences between the symmetry matched and the symmetry mismatched ones will be that are so far holding up that we have fewer more pronounced minima in the symmetry match case. So the next thing that Alexei has done is to put catalytic sites at the interface between the ring and the axle. And we can actually, and I'm not talking about today, but we've been doing a fair amount of enzyme design. So he can actually build active sites at the interface. And now what we're doing is using single molecule measurements to try and look at the relationship between fuel hydrolysis and rotational motion. And we have some tantalizing but rather preliminary results on that at this point. So just a couple other things. It's been we've been trying to engage the general public in protein design, and so we created this game called Fold It, where Fold It players start with a fully extended chain, and then they fold it up, and they can change the sequence. And they've been making all sorts of really cool looking proteins, and we've been making in the lab and solving structures, and a number of them actually fold up into very interesting new structures that are very close to what they designed. If I have I don't know, I'm probably way over time, but if I have a few minutes, I'll say a few words about how deep learning is affecting this. So let me just sort of conclude. I think I've shown you we understand the fundamentals of protein folding and assembly to the point where we can actually design brand new molecules and that do a wide variety of different things. It's just a super exciting time now because sort of like Manu alluded to, there are just so many different things that one can design. And so really excited about collaborations. If you have ideas, let me know. Yeah, I've realized that really the limitation now is more the network of people interested in testing things, or exploring things together. And again, the collaboration with Manu has been so wonderful. So, okay, let me just quickly go through acknowledgments. So the antiviral work done by Longjin and Brian and Ina, we've had really fabulous collaborators on the in vivo side. The diagnostic, the switch, the invention was that of Alfredo and Andy and Byung-ha. The antibody nanocages, that was work of Robbie Devine, a grad student in the lab, and David Wiesler solved the structures I showed you. And then I didn't actually get a chance to talk about extensible materials. The self-assembling 2D lattice work of Ariel Ben Sasson, again, that's been a fantastic collaboration with Joe and Manu. The pH-dependent fibers, the pH-dependent building block was built by Scott Boykin, and then Hao built it into the fibers, and again, another great collaboration with Joe and Manu. The de novo designed membrane proteins, Xianfu and Pei Long designed the alpha helical ones, and Anastasia and Samuel are designing the beta sheet ones. The calculations on the surface of cells for CAR T cell targeting, work of Mark and Scott and Jillian. That's been a great collaboration with Stan Riddell. Folded is the work of Brian Kopnick and a team of people now around the world. And Alexei and Yakov, really Alexei has been the driving force behind the whole rotary motor work. And do I have 2 minutes, Manu? Okay. All right. So now everything I've told you about is sort of based on the principles of physics. We're trying to design proteins that fold to the lowest energy states. But deep learning is changing this a little bit. So let me describe the basic concept. So we've been developing deep neural networks for predicting structure from amino acid sequence. And basically the way they work is you put in a sequence and they predict the set of distances between the amino acids from which we can generate a three-dimensional structure. Now, what we realized is that we could actually feed in completely random sequences. And of course, those are not predicted to fold to any structure at all. So you get a very blurry distance map. But then we could optimize the sequence to the point, basically optimize how sharp and contrasty the distance map was, to the point where it actually had an opinion about what it was going to fold to. And then we could generate structures. So you're probably familiar with the idea that if you have, say, something that can recognize images of cats on the internet, you can turn it around and start with and generate random images, and then tune them till the network thinks they look like cats, and those are called hallucinations. And in the same way, these are hallucinated proteins, because these aren't any particular protein. These are proteins that basically, these are sequences that the network thinks should fold. We didn't put any constraint on what that protein, what that structure actually was. We just said that just trying to optimize the sequence so that it will fold to something. And so this is just showing what the process looks like. We start off with a random sequence, and then we basically are doing Monte Carlo optimization of the sequence and just basically scoring how contrasty the distance map is. So you can see it starts off very fuzzy and blurry, and by the end of the optimization, we have these very strong contact patterns. So this is just residue by residue, and a dark spot means that the residues are close together. And when you generate models from these, you get these different types of structures. And if you do this many, many times, you get a really wide variety of sequences and structures. And what's interesting is if you take these sequences and fold them up using Rosetta, you get, they're predicted actually to fold to their design structures. This is completely orthogonal because Rosetta has this physical model, whereas the network really doesn't know anything about physics except what's sort of in its disembodied millions of parameters. So we've made quite a number of these proteins in the lab. They are they fold. They have the desired the target CD spectrum. They're very stable, and we've now solved structures of 3 of these by X-ray crystallography and NMR, and the structures are very close to the design models, showing that the network really can hallucinate brand new proteins that that fold as designed. So that's showing that this hallucination process can generate new proteins. There's some very nice properties here, even when you think about some from a more physical point of view. One of the problems with traditional Rosetta protein design is kind of myopic. It just sees the structure you're trying to design, and it basically searches for a sequence which is very low in energy in that structure. But as you do that, there's the possibility that there's some other state which is getting very low energy as well, and you only find that out after the fact when you have the sequence and you do a structure prediction calculation to see if your sequence actually folds to that structure. Now, in this, with the network, we can actually compute the probability that the sequence will fold to the structure of interest. And if we have a structure we want, we can then explicitly maximize that probability. And in that case, it's taking into account not only the structure we want, but also the whole landscape. So we can basically do landscape-aware sequence design. And the final example I'll give you is, so I told you about sort of just doing this free hallucination process. I told you about the very focused problem of you have a structure and you want a sequence which has very high probability to form to that structure. We can now make a hybrid of those two. We can say, look, we want this part of the structure to have this, to be in this state, for example, to bind a target, but the rest of it, we just want to be as simple as possible, and we want it to be such that the protein will fold. So we're keeping part of it a functional site fixed, and then hallucinating the rest of the sequence, sort of maximizing the probability that the protein fold to the state. So we've got basically a hybrid loss function. One part is just rewarding folding, and the other part is rewarding a recapitulation of that functional site. So here's an example. This is C3D. It's part of the complement cascade, and here are the 2 helices that bind complement are down here. And this just shows examples of what happens if you keep those parts fixed and hallucinate the rest. You get little proteins that where the backside is just buttressing the binding site, essentially. We can do this with insulin, and we can do the same thing with beta sheet proteins. And so it's kind of anyway, just. So anyway, so sorry, I'm probably gone way too long, but this was work of the free hallucinations work of Ian, Sergey, Sam, and Temuka. Chris and Basil really explored the sort of the idea of designing in the full energy landscape, and Doug and Sydney and Ju have been doing this constrained hallucination. So yeah, thanks for your attention, and sorry for going on and on.
Wow, thanks a lot. That was amazing. Actually, we have already many questions. So actually, there are a lot of people that raised their hands. So I think the first one was Lucas Carey. So Lucas, I'm allowing you to talk, so you can ask your question.
Oh, sorry, that was a mistake.
All right. Well then, sorry about that. Then I will move to Mutum Yaik Homba. And sorry if I'm butchering your name.
Hi, can you hear me?
Yep, we can.
Yeah.
Yes. Very nice talk, Dr. David. And so I just wanted to come to your question, your slides on the transmembrane helix design and the beta barrels. I mean, it's just more like a, so anyway, I'll just get off with the question. So how do you tune the polarity, in the sense how these transmembrane helices or beta barrels orient with respect to the cell membrane? In the sense whether they orient towards the periplasm, how do you tune that? And my second question is whether they pose a preference for the lipid environment that they are in.
Yeah, that's a very good question. So for the alpha helical proteins, it's been more straightforward than for the beta barrels. And for the alpha helical proteins, it's basically making the outside part that's in the membrane hydrophobic, and then we put snorkeling amino acids at the the membrane boundaries. But the beauty of de novo design is we can design for membranes of any thickness. They can be twice as thick or half as thick. We just change the lengths of the helices or the beta strands. And so we're very excited now about designing proteins for synthetic membranes. But yeah, I think you do have to keep in mind sort of the thickness and the properties of the bilayer that these proteins are ultimately going to go into.
All right. So we also have a question on the Q&A by Matthias Hohner that asks you, are these proteins immunogenic? And is it possible to create proteins in a way that is non or low immunogenic?
Yeah, so that's a really good question. I mean, so what I can say is that the binding proteins that I described that are 60 amino acids or less, they're very, very stable, and they're small, and they're very soluble. So far, we have not seen much sign of immunogenicity in animals. So for example, in the coronavirus therapeutic case, our collaborators did an experiment where over a 3-week interval, they gave the protein every 3 days. And then after 3 weeks, they gave the protein and then exposed the virus, and there was no reduction in efficacy. So there were no neutralizing antibodies against the protein that had been created. But of course, these haven't, we're going to find out actually in the next year, because we've started a couple of companies that are, putting de novo design proteins into people. But in general, our hypothesis is that, small, stable, and soluble proteins are not presented very well by dendritic cells. But one could go further, obviously removing T cell epitopes, B cell epitopes. The fact that they're smaller will make it easier. On the other hand, these nanoparticles are incredibly immunogenic. They are big, they look like viruses, and that's really part of the reason why these nanoparticle vaccines are so effective.
Jason Shane had a question. I will allow him to talk. Jason, can you hear us? Oh, all right. Well, so I will move to Buzz Baum had a question. So Buzz, I'm allowing you to talk. Oh.
Hello?
Buzz, yeah.
That was a totally amazing talk, David. I was just going to ask whether the fact that you can design all these proteins with specific structures, do we learn something about native proteins? So can you say, for example, can you predict how, say, COVID spike protein, why it evolved the mutations it did, and whether in the future can you predict its path of mutation in coming years, for example?
Yeah, that's a really interesting question. You know, I think we can certainly have a try at that, but one of the things that's perhaps I should have emphasized a little bit. One of the things I should also say is my talk was incredibly misleading because I showed you all these beautiful examples of designs that work. But of course, most designs either don't get expressed in bacteria or they end up as gunk at the bottom of your tube. So there was definitely some selection bias in what I told you about. But what we've also found is that, so when we're doing these design calculations, we're designing things to be in really deep energy minima, and that's how we're getting the accuracy. So, you know, the Rosetta force field does have, you know, it's certainly far from perfect. So there are probably errors on the order of a kcal per mole. And what that does is it makes it very hard to actually point to a native structure and say, well, this amino acid is contributing this. I think we could, so in terms of predicting the future course of coronavirus evolution, I think, you know, we could certainly say that substitutions that were likely to cause great destabilizations are unlikely to occur. But in terms of sort of the more subtle things that happen and sort of the reconfigurations that are likely to happen, you know, as new strains develop, you know, I think their models that incorporate, you know, past sequence information would certainly be necessary to supplement the physical model, the Rosetta physical model. Another example of that is, you know, directed evolution has been really powerful in terms of improving the activities of naturally occurring enzymes, more powerful than computational design, because you have to have really high, you know, the accuracy you need to predict, say, 5-fold changes in increases in activity is pretty small. Sorry, that was a long-winded answer to your.
It was brilliant. Thank you. Thank you.
But we're good at making completely new stuff. I mean, that's kind of the unintuitive thing. Making completely new stuff is actually easier than sort of, you know, making statements about, you know, the really complicated, delicate proteins that have come through natural evolution.
Right. So the next question is for Shu Han. I'm allowing you to talk.
Okay. Hi. Thanks, David, for the nice talk. So I just wonder, like, for when you screen through all these proteins scaffold, what's the library size do you screen through, and how many candidates do you have to screen to find sub-nanomolar affinity binder? And the second question is, what do you think about this rapidly developing phage display nanobody library, and what do you think you can replace phage display, and could this in silico design affinity mature those binders selected by phage display?
Yeah, well, yeah, so let's see. So the number of designs we actually tested in the different parts of my talk were very, very different. It has a lot to do with the size of the protein. So the reason why one of the reasons we focus on proteins with small sizes is we can actually encode each computational design in a single long oligonucleotide. And then we can actually test very large numbers of designs because we can get oligonucleotide chips from companies like Agilent or Twist, where they can encode up to 100,000 different oligonucleotides, each encoding a brand new different protein design. So for the coronavirus, that's what we did. We basically, on the computer, we designed 100,000 different proteins and we encoded them all in chips. And then we could use yeast display to identify the ones that worked the best. So on the other hand, for things like the transmembrane proteins, there we were just testing small handfuls of designs because those are bigger proteins, and we couldn't order lots of genes, and also the screening was much, much more difficult. So in the process, when I said we're going to now try and improve the computational design methods so that we don't need to screen so many, we have a lot of data now on the designs that work and the designs that don't work, and substitutions that change affinity, and we're trying to start doing machine learning on that to try and improve our methods. As far as phage display, so phage display is a very powerful method, but you can't specify what site you're going to bind to, and you can't really specify the properties. Well, you know it's going to be a nanobody, but you can't specify the stability of things. So I think that these computational methods will sort of take the place of sort of random library selection methods. It's sort of for the same reason that, like in modern technology now, if you want to build a building, it's like the phage display approach is you throw a big pile of bricks in the air and you let it fall and you keep doing it till it forms a nice building. But that's obviously kind of inefficient if you can just build the building from first principles in the first place.
So the next question is for Nina. I'm merging you, Nina. Yep.
100,000.
100,000.
What, sorry?
Nina, can you hear us?
Yep. Can
Okay.
you hear me? Can you hear me?
Yeah.
Yeah. Oh, fantastic talk, David. It's really mind-blowing. I'm really interested to know if you can design an intracellular thermometer. I'm particularly keen on doing this for long-lived cells like brain cells.
Well, that's a really interesting problem. We, yeah, just send me an email. If if you have a, that's exactly the kind of problem that we like to hear about. We just don't know what the problems are. So I think that would be a, let's discuss that. That sounds fun.
Brilliant. Great. Thank you.
So we have a question on the question Q&A. Somebody that so it's Zutao Yu that asks you, how about exploration on cellular delivery? Because obviously you are doing all those particles. The question is, can you deliver them?
Right. Well, we're working hard on that. I have a student who's who's figured out how to package guide RNAs inside the particles, and so we have, we actually have particles now that are built from those pH-dependent building blocks I told you about. So the particles come apart at low pH. The other thing is that when these trimers come apart, they actually permeabilize liposomes and cause their contents to be released. So we're now trying to put all the pieces together. And so the idea is that we, the particles get endocytosed. One of the formats, another format we've made actually pH-dependent plugs for those antibody cages. So anyway, the cage gets taken up, it comes apart at low pH, and then we have a guide RNA to do gene correction. So it hasn't quite worked yet, and if it had, I would have told you, but we're working hard on that. Yeah, so the idea is to get around viral vectors for delivery of biologics.
There's a question as well from Vad Perez that asks you whether it's possible to design self-catalytical reactions like the one that creates the chromophore in GFP.
Oh, yes. Well, in fact, Chunfu Zhu, who's the genius who designed the transmembrane proteins I told you about, he also has designed, as another collaboration with Manu and Joe, he's designed proteins which bind to chromophores which fluoresce in the far infrared for deep tissue imaging. But he has also designed proteins which do the GFP chemistry. Wow. So he actually has completely synthetic all helical GFPs now.
But the question is, do you think you can then do, like predict complete, you could do completely different chromophores from the ones that are, you think you can do that?
I think there it becomes sort of a chemistry question, we need collaborators who can say, well, if you could bring, it also has to be cases where the side chains, they have to be reactive, and but yeah, if you can tell us that, if you can say, if you bring these three side chains together in this geometry, this reaction will likely take place and that will be fluorogenic. I think that's totally doable. We just need marching orders.
Yeah. All right. Sorry, let me just go back to that. Henry Wood had a question. Henry, I'm allowing you to talk. Oh.
Okay. Yeah. Can you hear me?
Yep.
Yeah.
Great talk. Thank you, David. Just a bit of a broader question. You've obviously got a lot of programming experience. What do you think the main obstacles are to molecular biology reaching a point where you can easily program it from a computer? Or is that maybe the wrong way of thinking about it?
No, I think that's a reasonable way to think about it. Biology is really complicated. Everything I talked about was with single protein systems, and already, we're really stretching the limits. The amount of computing power that went into a lot of the calculations is very large. So for example, one of the things that we do before we order designs is we take the sequences and we send them out to a distributed computing project that we started a number of years ago called Rosetta at Home. And basically people on their on their home computers basically predict the structure, and they send us those predicted structures back. And so we can check in that way that the sequence actually is predicted to fold to that design structure. That's a big calculation. So if you're now going to be programming molecular biology, it's going to be hard to do it at that level of detail, because you'll now need to be simulating not just individual proteins, but whole networks of proteins, and maybe their cellular membranes as well. So you have to go to higher levels of abstraction, and the key for any kind of computational model is that you need to be at high enough resolution so that your calculations are accurate, but they still have to be computationally tractable. So, you have to be able to do enough sampling so that you're really figuring out how your system works, at least according to your computational model. I think for higher level questions in biology, I think that's just very hard right now. I think we'll get there, but it's going to require, much more powerful, larger scale computations, and probably new types of models.
All right, so in the interest of time, I will ask the last question. It's from Himami Tendon from the LMB. He asks, while designing small proteins in Novo, how do you computationally filter out those proteins which can be very good as binders, but can have non-specific binding or off targets?
Yeah, well, we do that sort of what we call heuristic negative design. So the one, like you could imagine taking each design and then docking it against every possible every host protein, and throwing out those that are predicted to bind other targets. But obviously that's, it's an example of something that's not really feasible. So instead, we try and build in properties that are going to likely to give rise to specificity. And the most obvious one is we avoid big hydrophobic patches, which are likely to lead to non-specific binding. So we're sort of restricting the number of hydrophobic residues that be close together, and then always trying to make sure we have a lot of polar interactions with the target as well. And when we do that, we have a number of proteins where we've done sort of whole cell pull downs, and we only pull down the desired target. So I guess the answer is by getting very high stability, and then by going for more polar interfaces. And you saw, I showed that briefly, what the surface views look like. That's how we aim for specificity.
All right. And you think the avidity helps? Was that sometimes? Oh, yeah.
Yeah. So the avidity, the avidity is certainly a way of getting, in the case of the coronavirus, we didn't need better than picomolar binding to neutralize any given virus. But once it starts to mutate, if you're at 10 femtomolar, you can lose 3 logs of binding affinity and still neutralize. So yeah, so that definitely gives me more.
All right, so in the interest of time, I will stop the discussion here because you have a very tight schedule now. So we'd like to thank you again very much, David. That was an absolutely fantastic talk. Thank you all the attendees. I think we maxed out Zoom, the Zoom account of the LMB. So great, and then so David, now you should join.
Well, thanks for all the great questions. I wish we had more time to talk, but if people would like to collaborate, please send me an email. We're really excited to explore new areas.
Thank you very much.
So Manu, what do I do now?
So now you just go to the other link, which is